I think you have to sort of go back to first principles, and you have to, when we have small molecules, it's very easy. We make a generic, it's exactly the same. Because biological molecules are complicated, you cannot say that it is exactly the same. So what the EMA, the MHRA, the regulatory bodies have done is set up a really rigorous process for getting a label for a biosimilar, getting a license. So they have to go through extensive clinical trials that look at the pharmacokinetics, that look at PK values. They then undergo an exploratory clinical trial in a comparative population to compare to the originator. So you will get you will compare the safety profile, you will compare the outcomes, and really the clinical trial is almost the least important bit of it. The main work are these sort of pharmacokinetic studies where you essentially get graphs that overlap and that show that the molecule, the efficacy and how it works in the body is exactly the same, with the small scale studies to prove concept in reality. And I think I'll come back to the point I made about sort of as the manufacturing process goes for biologics, they evolve over time. So changes in the manufacturing process, and I believe there's something like 23, it might not be the exact figure, but around about that, changes to the manufacturing process of Rituximab. So the molecule has changed a lot, but we still get the same results. I mean, and this is important because Rituximab has been, alongside Imatinib, they're probably two of the best drugs that we've had in oncology and haematology. They're very effective treatments. So we do need that reassurance, and I do believe it is there. So I think clinicians need to, there are plenty of papers understanding the evidence, but I think taking reassurance from the fact that, something like NICE, NHS England, all the regulatory bodies have accepted these medicines work, and you don't need to go and repeat the phase 3 trials. To get the benefit of these, we need to use them. We don't need to be waiting for trials. One thing I discovered when I was learning more about biosimilars was actually the term biosimilars was sort of an invented term, because people knew that they were the same molecule, but we couldn't really describe them exactly the same. So I think it's getting your head around that. There are a couple of considerations for clinicians though. One of them is the pharmacovigilance. So because they are essentially similar and therapeutically equivalent but not the same, you do need to be able to know exactly what it is you're prescribing. So clinicians won't be, the medicine won't be changed. You won't prescribe as a clinician Rituximab and not know what brand you will get. And in fact, you will have to prescribe the originator molecule by its brand name as well now. So branded prescribing, so prescribing something by brand, and also tracking the batch numbers. And pharmacy systems will track the batch numbers. So there is that additional layer of sort of safety monitoring, pharmacovigilance that will happen with these new medicines. In addition to that, with all new medicines, which they are called black triangle medicines and are monitored. So I think there's reassurance there that we knowing that we're monitoring them, knowing we're looking at them, knowing we need to gain experience.